Good afternoon, everyone, and welcome to the second part of this year's virtual Soapbox Science Milton Keynes event. I'm Dr. Barbara Kunz from the Open University, and I'm going to lead you through this afternoon's session. Soapbox Science has been created as an outreach platform to bring science to the street and general public, as well as to highlight the work that women and non-binary scientists in STEM do. We're very excited to have the second part of our 12 amazing scientists speaking today and telling you about the research they're doing. If you have missed the first part of our event on Thursday, there's a recording of that session on YouTube and Facebook, so make sure to check that out. Each speaker will have 5 minutes today to give a short presentation introducing themselves and their work. This is followed by 5 minutes time for questions. So if you do have any questions for a speaker, please make sure to type this into the chat and comment. And they are going to pick, or we are going to pick this up during the Q&A. There might not be enough time to answer all of the questions, or you maybe have a question after the time slot of the speaker. But don't worry, just type in your question. Make sure that you indicate to who the question is for, and we will pass this on. And we will also pass on all the not answered questions, and the speakers, they will publish a blog post where they will talk about where they answer all of the questions. We'll publish this on our homepage, and you can see the link below the video, and we will also announce this on our social media channels. So let's get started. And today we will start with Dr. Navya Thomas, a researcher in membrane technology at Cranfield University.
Thanks, Barbara, for that introduction. And good afternoon, everyone. I'm really excited to kickstart the day 2 of Milton Keynes Soapbox Science event this year. My name is Dr. Navya Thomas, and today I'm here to talk to you about 3D printing, membrane technology, and clean water production. One of the many ways that we transform contaminated source water, be it groundwater, surface water, wastewater, or salty seawater, to clean water is by the use of membrane technology. Today, the global membrane technology market is valued at $18 billion US. Now, a membrane looks like this sheet of paper, and it contains millions of small pores that are invisible to the naked eye. But these small pores selectively retain contaminants while allowing clean water molecules to pass through. At a membrane treatment facility, after hours and days of operation, a clean membrane like this gets transformed to something like this. And what we see here is the accumulation of contaminants on the surface of the membrane, which is known as membrane fouling. Now, membrane fouling is undesirable because by blocking the membrane pores, they are rendered inaccessible for clean water production, reducing the water production capacity of the membrane process. You have a higher pumping energy cost because you need a much higher pump pressure to pump against a blocked and fouled membrane. At an operation facility, you have to intermittently stop the plant, clean the membranes with chemicals, and then resume operation again. And all of this adds to the cost of clean water production. One of the culprits for membrane fouling is identified to be the spacer. Now, a commercial spacer looks something like this, and it has a geometry resembling that of a fishing net. Now, a spacer is used in a commercial membrane assembly because it helps to keep apart two membrane flat sheets, and the presence of the spacer between the membranes allows the waters to pass over the membrane, creating a flow channel. It provides mechanical integrity to the fragile membranes against the high pressure of the feed water, and it also acts to an extent as a turbulence promoter to reduce membrane fouling. But in reality, what we have observed is that these fouling patterns trace the contact points of the spacer on the membrane itself, indicating that the geometry of the spacer influences membrane fouling. In my research, I looked at how we could think outside the box to redesign spacer geometry, and the solution to that is 3D printing. With the power of 3D printing, we can transform a two-dimensional spacer geometry, which looks like this, to a more complex, intricate, mathematically derived complex geometry, which looks like this, which is three-dimensional in nature. And these geometries cannot be created with the conventional manufacturing technology, and thus we need the power of 3D printing. The beauty of these complicated geometries are twofold. One is that they're perfectly curved at any given point, which means that they minimize the straight edges that you see in a conventional design, and thus reduce the propensity for the foulants to accumulate on the membrane surface. They also have this interpenetrating maze-like structure. So when the water flows through these complex geometries, they're way more turbulent when compared to how the water would flow over a conventional fishing net geometry. So in our tests, we transform these three-dimensional geometry to that of a spacer geometry, which looks like this. And we then test them against the performance of a conventional spacer design for membrane processes. And what we have observed is that by using these 3D printed spacers, we not only reduce membrane fouling by about 50%, but we also have the added advantage of increasing the clean water production by 60%. And that is a win-win situation. The use of 3D printing for spacer design is just the tip of the iceberg. Today, researchers are looking at how we can use 3D printing to 3D print the membranes themselves with much more precision of pore control to create high performance membranes. So that's how we can use 3D printing as an ally to create improved membrane processes and improve clean water production at a global scale. That's it for me, and thank you for listening.
Great, thanks, Navya. That sounds like a really interesting, fascinating thing to do with 3D printing. Never thought of that. Thanks for your presentation, and I'm now going to hand over to Sophia to ask some questions.
Thank you, Barbara, and thanks a lot, Navya, for that really interesting talk. The first question is, these 3D materials that you are developing, in what sort of water filtration systems would we normally find them? Are they kind of for community water supplies, or are they for smaller things like air conditioning units or boiler systems?
So we've tested them for across all scales. So the membrane technology itself, you can divide them into different membrane processes depending on the pores that you use. So you have microfiltration, ultrafiltration, or reverse osmosis for seawater treatment applications. And these are designed for large-scale membrane applications. That's the final objective, where you would have membranes in a commercial setup, and you would also have spacers that are used at a commercial scale.
Okay, that's great. And these 3D printed materials you design, are they already available in the market? Or if not, how soon do you think we could see them in a commercial setup?
That's an interesting question. So we have filed a patent for these designs, and we have interest expressed by commercial entities who want to test them at a commercial scale. So all the tests that we've done so far are at a lab scale. So the natural progression would be now to do a pilot scale study and then a commercial scale study. So hopefully in the coming years, we should have a viable commercial product that's cost effective as well.
Well, looking forward to that. So are they quite durable, these new materials, compared to what we normally use now?
Right. So again, it boils down to the choice of materials. Today, we're talking about a printing technology where you can choose from an array of 3D printing materials. So when we started off initially, we did hit a rock bottom because the choice of material that we had made would just integrate the spacer under high pressure. And then we had to go in and try and use polyamide, which is nylon, generally known as nylon. And that was able to withstand the high pressure for membrane processes. So today, you can choose from ceramic, metals, or plastic material to make these spacers, or 3D printed material for that matter. So depending on the choice of printing technology, the application, you can definitely come up with something that's durable and long-term.
Brilliant. We get some comments on Facebook. People find it, Katrina says, it's really fascinating, all this science. What I'm interested in, all these, because you use different geometries for all these matrices you design. Where do you get the inspiration for all these different geometries?
Oh, okay. I'm really happy that you asked that question. So interestingly, these are mathematical geometries, and it has been known to the scientific community since the late 1800s, but manifesting those designs was not possible by conventional manufacturing technologies. And that's where we realized the power of 3D printing, because it's extremely complex. and you can't create them with the conventional manufacturing technology. So we have more than 40 different shapes, and researchers across the world are even trying different other designs. So you have somebody who's tested honeycomb structure, somebody who's taken inspiration from the aerodynamic wing design of a plane or wave structure. So, and that's the beauty of 3D printing, anything that you can think and imagine, you can create and prototype and test it for different applications.
That's great. And one last quick question.
Sure.
Are these 3D, is 3D printing expensive? Would it increase the cost of these filtration systems?
Right. So that is a touchy subject because a 3D printed spacer would now cost about 10 times more expensive than a commercial conventional spacer. But just to give you a background, in 1980s to print a material, which would take about 11.5 hours, today we can do that under 6 minutes. So that just shows the growth of 3D printing and the advancement in the technology. So going forward, I think that we could match up to a cost-effective solution, but it will take some time.
That's great. Thank you very much, Navya.
Thank you, Sophia.
Great. Thanks for taking the time to answer the questions and your presentation. And now we're going to have our second speaker, Dr. Chitra Balakrishna. She's an educator and researcher in cybersecurity at the Open University. Over to you.
Hi, good afternoon, everyone. I am Chitra Balakrishna, and I'm an educator and a researcher in internet technologies and cybersecurity for over 20 years now. I work on technologies that enable computers to connect and talk to each other. But today's internet not just connects computers, but it can connect people, information, places, and even objects. Like, for instance, this smartwatch of mine talks to my mobile phone to say how long I slept last night. Or my mobile phone talks to my smart light and turns it on every morning when I wake up. While we have begun to conduct our entire lives on the internet, right from exchanging money, doing shopping, learning, entertainment, and even trusting it with the most intimate aspects of our lives, we've also begun to face some of the dark consequences of the internet, such as the ransomware attacks, cyber crimes, falling victim to online scams, etc. In today's digital world, knowing what to do and doing what it takes to protect your information and yourself is not an option anymore, be it a small company, a large organization, a common person, or even your 8-year-old who spends a fair amount of time on the internet. So cybersecurity today touches upon every person's life. However, cybersecurity as a concept is often viewed as being extremely complex, extremely technical, geeky, and even scary for some people. And this has led to a severe cybersecurity skills shortage. Would you believe me if I said there are nearly 4 million cybersecurity jobs that are not filled each year globally? Yes. So there is a dire need to change the way we teach cybersecurity. There is an urgent need to demystify cybersecurity for the larger population. Now, how do we teach cybersecurity, or for that matter, anything in our universities today? Like this. Sorry, like this, actually, right? In lecture halls and lecture theaters. And guess how we taught our students 100 years ago? Like this, in lecture theaters and lecture halls. The world has changed dramatically over the last 10 years, yet the way we teach has not changed much at all. It is this that has fueled my passion and research, especially during the last few years now. Cybersecurity is not just about knowing what to do, but actually doing the right thing every single time, even if it means modifying your behavior. I am inspired by Diane Ackerman's quote that says, "Play is the brain's natural way or favorite way of learning." Research shows that games influence human behavior much more than any book or instructions. Now, how do we learn some of the most basic life skills? For instance, swimming. Not by reading about it, not by listening to a lecturer or an expert talk about swimming, but actually doing it by actually swimming. And what motivates young children to learn to swim? They don't want to learn to swim simply because it's an important life skill to have. They want to swim because of the sheer joy of swimming and being in water. The same should apply for learning. It should be, learning should be done for the sheer joy of learning, and the skills and the knowledge you would acquire should merely be a natural consequence of that joyful learning activity. In my work over the past 5 years, I have developed cybersecurity games to teach ethical hacking techniques for 20-year-olds at the university. I've also developed games to teach basic cybersecurity skills to adult learners aged between 40 to 65 years with no formal degree or qualification, with no digital experience or skills. And learners from across these age group have vouched that game-based learning is not just enjoyable, but is highly engaging. They've even gone on to claim that they retain what they've learned better by playing the games than they would have by reading books. Games are indeed the new books. The games that I have developed have a story-based narrative, a role play, strategy, and all of these impart the learners risk-taking, persistence, attention to detail, problem-solving. All of these behaviors are ideally suited for effective cybersecurity training. Last but not the least, game-based learning offers the learners a safe place to learn by trial and error, where they are not penalized for failing, where they can learn without the fear of failure, where every learner has the opportunity to experience success, the joy of success, which our current education methods unfortunately don't offer. That's it from me. Thank you for listening, and here's to joyful learning.
Indeed, thank you. That was really fascinating and I think really important. I'm sure there are like lots of questions.
Yes, they are. So thanks a lot, Chitra. I'm sure we're gonna start getting lots of questions both from Facebook and YouTube, but I'm gonna start with, is there a risk that, when we think of games, that learners might not take it seriously, take the learning seriously, because we don't take games that seriously, we focus more on the fun of it? So is there such a risk, especially with such complex topics like cybersecurity?
Yes, I think it eventually boils down to the mindset. Obviously, we can't break down the current teaching and education system and build it from scratch. We have to build from where we are now. It's a basic requirement of change of mindset. It's always, ingrained in us that learning is not fun, or if you're having fun, you're not learning. Somehow don't fit in the same situation or sentence, even for that matter. So it's a change of mindset, A. Two, I think it's about offering alternate means and channels for people to learn. If there are learners who are better at learning in traditional methods and means, we already have, plenty of resources to offer, cater to them. It's about those learners who have an entry barrier in learning through conventional methods. We need to get them on board, and the digital natives, as we call them, the millennials who are exposed to digital media and multimedia right from when they were born, have their brains wired differently. They are triggered by multimedia, not in a singular teacher-focused lecture-based medium of instruction. Wouldn't possibly work for those digital natives who are exposed to multimedia and learning from a variety of simultaneous medium. So I think it's a matter of offering an alternate medium and breaking those entry barriers for those learners who may not be able to effectively learn using a traditional conventional method.
That's really interesting. So based on your experience and your research, when developing such a game, what is more important, appealing to the player or make sure there's an effective teaching there?
That is the key challenge of any game-based learning methodology. Making sure the learning intent is the core of the game is key. And then adding the game mechanics or gameplay strategy around the core learning intent, is a challenging task. It's, there is a curve to be able to create a simple learning game. There's a lot of design challenges. There's a lot of interaction challenges that needs to be overcome to, ideally create an effective game-based learning. we are a long way. There's plenty of research. The work that's happened in the last 10 years is only a tip of the iceberg. There's plenty of work, both from behavioral scientists, psychologists, artificial intelligence experts. They could all come together to make learning more natural, intuitive, and joyful, and offer that intrinsic motivation to learn, and break those barriers for learning. So it's still a very early stages, but yeah, keeping the learning intent at the heart of any game-based learning is the key.
That's great. And I have a final question. With technology rapidly advancing, and of course we get new cybersecurity threats all the time, and fraudsters, keep coming up with new ideas all the time on how to trick us. How easy it is to adapt these games and kind of update them as we go along to make sure we are up to date with the latest threats?
Absolutely, that is going to be there is a challenge in terms of time-related challenge in keeping the content up to date in such a fast pace cybersecurity world. However, if you're training cybersecurity professionals, the techniques and tools are still the same. There may be various challenges, but the tools and techniques, the underlying principles, cybersecurity principles, still remain the same. So if you're training cybersecurity professionals, games are a great way of training them, particularly for ethical hacking techniques, where we need a safe place to learn those hacking techniques, to think like an attacker, to be able to defend and protect from those attacks. It really works in those areas. But if you're talking about awareness creation, security awareness creation, again, there needs to be, because there are new threats and risks happening each day, new attacks emerging each day. So you need to adapt the games, to such a fast pacing, that is a challenge, yes.
Fantastic, fascinating. We all need to play more then. So,
yes.
Thanks a lot, Chitra. Great stuff.
Yeah, thank you. I'm sure we could fill like a whole hour, but I'm afraid we have to move on. But if there have any been questions that haven't been answered, then Chitra will take the time and answer them, and we will publish that in a blog post. So thank you. Thanks a lot for that great talk. And now we have our next speaker, Amy Withers. She's a PhD researcher in insect molecular biology at Lancaster University and Rothamsted Research.
Hi everyone, I'm here today to talk a little bit about some of my PhD research, which has used DNA and RNA to learn more about crop pests. So crop pests are a real global problem. They can result in millions of tons of food to be lost and huge economic burdens as well. And we're interested in one crop pest in particular, which is called the fall armyworm. And despite what its name suggests, this is actually a type of moth. And you might be thinking, this is quite a small moth, and how can this small moth cause so much damage and become such a global problem? And the reason for that is that because when it is a caterpillar, it will eat millions of tons of food. For example, in Africa, it's expected to lose up to 20 million of maize per year from this small caterpillar. And this is particularly interesting because the fall armyworm is actually a type of invasive crop pest. And this means that it's new in Africa. So before 2016 in the Americas, it was a known crop pest. However, in 2016, it reached Africa for the very first time. And since then, it has rapidly spread across the whole continent. And then it also spread into Asia and Australasia as well, which is why it's now considered quite a global problem. And one of the reasons it spread so quickly is through rapid flight. So once the moths have emerged, they can actually fly up to 300 miles before they lay their eggs. And about 300 miles is the distance from London right up to the Scottish border. So you can see how quickly this can become problematic and cause such a spread. And one of the reasons we wanted to know about this spread is because of things like insecticide resistance. Because as these populations are mixing, this can lead to the spread of insecticide resistance across the continent. And this means that when the farmers try to protect their crops, they won't be able to, and the insect will be able to survive. So one way we look at population mixing is by using DNA. And DNA looks a little bit like this. And this is known as the instructions of life that is made up of different building blocks. And this is because there's 4 bases, which are called A, T, C, and G. And it's the order of these bases that can tell us so much about the species. And the bit we're interested in particular are called microsatellites. And these are really variable regions that allow us to identify different populations or even different individuals. It's the same sort of technique that forensics will use when they're trying to identify criminals at crime scenes. However, we used it to identify which populations were mixing with others across the African continent. And we did this by collecting samples from 6 different countries. And then we were able to use this DNA to tell us how these populations were mixing. And we actually found out that they were very regularly undertaking long distance flights across the continent, meaning they were mixing really well across the country. And this would mean that as well as between countries, across different countries as well, if they did have insecticide resistance, this would spread very quickly. So while insecticide resistance is a problem, there are ways around it. And one of these is using things like biocontrol. And we're particularly interested in microbial biocontrol. And this is when you can use a virus to target the crop pest, and this can lead to the crop pest dying and the crop is protected. And these are really useful because they don't harm any other species. So things that we want there, like pollinators or natural enemies, are unharmed. And it is only the crop pest that is actually affected. However, occasionally the crop pest won't receive a high enough dose of the virus, and this means it will survive, but still be carrying that viral infection. And we wanted to know a little bit more about what effects this viral infection can actually have on the adults, because we know it hasn't killed them, but is it having any other effects? For example, is it changing how they fly? Is it changing their immune system or energy metabolism? And these can all be really important because it can help us get an understanding of why they're spreading so far. And to do this, we used RNA. So RNA is basically the messenger from DNA to the cell machinery that produces things like proteins. And what we're able to do in the lab was fly the moths on specialised machinery that allows us to record the distance and the speed flown. And then we extract this RNA to get a live snapshot of what was happening in the moths' bodies during this really high energy demanding activity of flight. And we found that if they had this infection present, they were actually needing to use a lot of genes related to immunity, and they were still trying to sort of fight this infection. However, this came at a cost because those that had the infection also flew shorter distances at slower speeds. And this can have important implications because now we know that if they do survive those infections, it might still affect how far they're actually able to spread. So overall, by using DNA and RNA, we have been able to learn so much more about this crop pest. And I'd just like to thank you all for listening to my talk today. And are there any questions from anybody?
Thank you, Amy. I'm sure there are questions. That sounds like really fun experiments and really interesting lab work. I'm really curious who asks what questions.
Absolutely. So I think, my first question has to be, do you have these moths in the lab here at Rothamsted? Did you bring them all the way from Africa?
Yes, so we have got some of the moths in the lab that we work with. They're kept under very strict conditions, so there's no risk of them ever escaping, so please don't worry. But yeah, we do keep a culture of them in the lab, and at any one time we can have 100 or so. These are really useful for experiments because it means that we can study things like flight, which in the field obviously really hard to do because they're so small, you can't really track them, especially when they're flying such huge distances as well. So having that lab culture is really useful to be able to let us learn more about them.
Absolutely, yeah. There is a question on Facebook from Luca, who says, by the way, it's really interesting talk, Amy. How can we stop pests like this from spreading in the first place?
So one of the main ways that often spread is by doing things like checks when fruit and vegetables being imported. So we do try and mitigate them actually becoming established, and we do try and keep an eye out for them. However, unfortunately, they do sometimes manage to reach a new place. And this is particularly problematic. So it's likely that from America to Africa, it was introduced through something like a plane or a boat being involved. But once it was actually in Africa, because it can undergo such long distance flights, it's very hard to stop that because they're in the country, they can cross borders easily. And that does lead to quite rapid spread.
Yeah, they seem to fly long distances, don't they?
Yeah, they do.
So, I mean, you're working on trying to understand them and develop more effective biocontrol strategies. What are the challenges of that?
So some of the challenges is more just trying to understand the actual biology of the pest, so learning what sort of doses are needed for the biocontrol to be effective, and also trying to make sure that biocontrol agent is definitely very species specific, so they have to go through quite strict protocols to make sure that non-target species are unharmed. And this is why the things like the viruses I study are quite important, because to date they are really species specific. And it's been shown in field experiments that when they have been used, things like pollinators and natural enemies weren't affected at all. And it was just that crop pest that was at a lower prevalence by the end.
Right, great. There's a question coming from the YouTube. So say, is this use of RNA to study pathogens new, or is it new for this species? And could the benefits, what are the benefits of using this RNA approach rather than compared to just studying the behavior of the moths?
So by using the RNA, we're able to sort of learn a lot more about the molecular biology, so actually see what's going on that otherwise we can't see. So for example, the RNA showed us that they were still actually upregulating the immune genes, so they were still fighting this infection, even though before we thought that maybe it was just being carried but wasn't actually needing to be fought or anything. And these are also techniques that are really widespread, so they're not just used for the fall armorworm or in insects, they can be used in any species, which makes them really useful as well, because even though I've done this work in insects, the same experiment could be carried out in a mammal or a bird even.
That's great stuff, Amy. Thank you very much. Don't know whether we have more time, Barbara.
Yeah, I think we have to move on to the next speaker. But if there, again, we will get to all of the questions, or the speakers will get to all of the questions. And next we will have Alina Igna, a PhD researcher in molecular plant pathology at Rothamsted Research.
Good afternoon, everyone. I would like to start my talk by reminding you of your recent feeling when you went shopping and faced a serious shortage in bread, pasta, and flour. For me personally, this was a very frustrating situation. But I would like to go even further and remind you that all those prime ingredients are produced from wheat, which is a cereal grain, and that a disease of wheat could cause a similar shortage in our supermarket shelves. This is a very worrying fact when we are actually facing an ever-growing human population. Therefore, we need to protect our crops from diseases so we could feed the population. In the case of wheat, one of the most devastating diseases is caused by Zymoseptoria triticale, or for short, Zymo. Zymo is a fungus, and when I say fungus, think of a mold which infects the wheat leaves. Now, we all learned that a green leaf like this one can use the sunlight to make nutrients for the plant, and we call it photosynthesis. But Zymo causes this type of lesions on the surface of leaves, which reduces the photosynthetic capacity of wheat. And this could cause up to 50% yield losses. Currently, farmers across UK and Europe use almost 70% fungicide applications annually to combat this disease. But sadly, Zymo started gaining resistance to this type of chemicals. With these worrying facts in mind, we need novel ways to try help wheat fight off Zymo. And this is where my PhD research work comes in. I am using a plant called tobacco, but not the tobacco that you smoke, which recognizes Zymo. And I want to fish out the immune components from it, which recognizes Zymo. But before we dive in into the strategy that I am using to do so, I would like to highlight some important aspects. When we do have a microbe that is causing a disease, we call it pathogen. In our case, Zymo is a pathogen. Now, this pathogen, in order to infect the plant, most of the times, it uses some tiny molecular components, which are called effectors. But on the other side, the plant can detect these effectors through immune components, which are called receptors. So effectors are bad, receptors are good, and receptors can detect the effectors. Okay, but now something more interesting happens in a plant. The plant receptor sometimes needs a body to form a complex to fully make the plant aware of the infection. And the main difference between a body and a receptor is that the body has this wiring system, I think of a conductivity, which can send a signal to the plant saying, hey, we are infected, start defense response now. So how can I use this knowledge to identify an immune receptor recognizing zymo? Well, I can basically use the body by making it my bait, and I'm adding a yellow tag to it, just like this one. Now, in a real scenario, what happens is that the tobacco plant has an unknown receptor that recognizes the zymo effectors, just like that. And upon recognition, the body comes in to help send the signal to the plant and install the defense response. And this is the moment when I come in with my fishing rod. And I'm using this yellow bit to attach to the yellow bits of my bait of the body. And this is how I can scoop it out as a whole complex. So basically, this is how I use the strategy to fish out immune receptors that recognizes zymo. And I can now be able to potentially help with by, for example, burying him the receptor, or I could even better, perhaps expand this topic in the upcoming question session. Thank you so much for listening, and I'm happy to answer any of your questions now.
Thank you. That sounds like really vital research you're doing there to keep us in pasta and bread and cake. I'm sure people have questions for you.
Yes,
Sure.
we have a lot of questions. Thanks, Alina. It was really interesting, but as you said, maybe we can expand a little bit more in the questions that people are sending. The first one is, if this pathogen, this fungus, is it a global problem, or is it more focused maybe in the UK or some other places in the world?
Well, I could say it's global, but it's predominant in the UK and Europe, because also it relies on geographically aspects and also the climate conditions, such as humid, kind of like when you have autumn or cold winter, kind of like especially wet winter, such as in UK, Germany, France. So we actually have more problems concentrated in these parts of the globe.
That's great. Thank you. We have a question from Facebook from Luca that is very encouraging. He said he loves the models, and he's asking, what part of zymo do you expect the immune receptors to recognize and bind to?
So the immune receptor from the tobacco plant that I'm using, this will require a little bit of background, but I will try to be super quick. In plants, most of the time, we have a receptor that already, I think it's still on. So basically in plants, we either have a lonely receptor that will need a body, but sometimes what happens is that this wiring, basically, if I take it from here, oops, I just run it and I attach it here. We could also have receptors that they already have in the wiring. So it could already by itself detect and then send a signal by itself. So basically it could do two jobs in one. But in the case of my research project, because I'm using this molecular bait, is because the research has found that without this body inside the plant, the immune response cannot actually be triggered. So basically, that's how I know that I would expect a receptor that it's in need of the body because he cannot have the wiring by himself. I hope I answered the question.
I think so. I think it was very clear. I think the models are really helping. There are also questions from YouTube. So I'm just gonna fire it away. How many immune components from the tobacco plant are needed to be transferred to give the most resistance response of wheat to zymo? Is one enough?
That is a good question. Okay. Could I make a big example? We have this pandemic. We have the COVID. You get just one injection is not enough. You need a second, and then you need a booster. So it's pretty much, I would suspect, most probably, this is what happens also in immunity in plants. They would need not just one receptor, they need 1, 2, 3, and maybe we're gonna be an army. But if I could go even deeper into that, in tobacco at the moment, my research project is focusing at the moment on the strategy. And I could scoop out one receptor. That could actually reduce potentially could reduce the virulence. When I say virulence, I mean how bad the pathogen can infect the plant. So it can reduce that by a small percentage, but not necessarily make it resistant, I mean, the plant. So the research has already found, just to give you an idea in numbers, 63 candidate effectors, but only a couple of them, like dozens of them, are relying on this kind of strategy that I'm using on this body system. So ideally would be all effectors, all in.
Great. There are some more questions, but I think we need to move on. So we will try to answer those in a blog later on. So thank you.
I will definitely.
And over
thank
to
you,
Barbara.
Beautiful.
Yes, thanks for this. Great. I really love the models too. Next up, we will have Katherine Gobey. She's a PhD researcher in plasma processing at Cranfield University.
Yeah, thank you very much. My name's Katherine. I am losing my voice a little, so hopefully you can all still understand me. So I'm a PhD student at Cranfield University and I study plasma. So firstly, what is plasma? Plasma is actually the most abundant form of matter in the universe. So it's the fourth form of matter. You have solid, liquid, gas, and then plasma. So as I said, the most abundant form of matter in the universe. Most of what's in space is made of plasma. So the butterfly nebula behind me, that's all plasma. On Earth, you're most likely to see plasma as lightning, which is a plasma made of air. And you may also be familiar with these things, plasma balls. So let's turn this on and get the lights off. So in here, you'll have a gas, which is probably going to be neon. And there's, can you see this okay? So there's an electrode as well. So when the plasma ball is turned on, the energy from the electrode gives lots of energy to the atoms that are in that ball. So what happens then is the atoms of gas, so usually you have electrons that are orbiting around a nucleus. So if you put enough energy into these electrons, they actually get so much energy that they can separate themselves from the nucleus. So then in a plasma, it's actually overall a neutral charge, but it's full of lots of negatively charged electrons and positively charged ions that have lost some or sometimes even all of their electrons. So in plasmas, you also get a lot of radiation, so visible light, which I think you can just about see here, and also lots of UV as well sometimes. And that happens when the electrons that are whizzing around on their own, they fall back into an atom, and then they release that excess energy that they had as the radiation. So because of all of those features, plasma has some really exciting science, and it means it can be used in lots of applications. So my project focuses on plasma cleaning. So I use plasma cleaning in the concept of concentrating solar power mirrors. So concentrating solar power is a method of renewable energy generation where you have lots of massive mirrors that focus the sun's light onto a central receiving unit that gets very hot, and you can generate electricity with that heat. So these tend to be out in the desert because there's lots of sunlight in the desert, but there's also lots of sand and dust in the desert. So those mirrors get coated in sand and dust, and then they don't work as well as they should because it doesn't reflect as much light. So at the moment, what they do is they use big trucks and they just blast those of water and brushes onto the mirrors, much like you'd wash your car. But in the desert, water is a really precious resource. So it can be very expensive to buy water in these places. And it's also just ethically not that great because water should be going to people rather than things like this. So I'm hoping to use plasma to reduce the amount of water that they need to clean these mirrors. So plasma cleaning is already quite an established technique. It's used in lots of different industries already. It works in a number of different ways. So for my application, it works firstly by breaking apart the soiling itself. So I mentioned that the plasma contains lots of reactive species, and these don't want to be in their reactive states. They want to be stable and bound in a neutral state. So if your charged or reactive species in the plasma hit the soiling on that mirror, it can react with that or latch onto it, and it can dislodge it from the mirror as it does so. Plasma can also interact with the bonds between the sand and the mirror itself. So for example, the charged particles might overcome the electrostatic bonds between those two. So plasma also is really great at something called surface energy modification, which basically changes the wettability, so how much water loving or hating a surface is. So my plasma makes the mirror surfaces hydrophilic, water loving, and it does this by the high energy electrons hit the surface and they break apart the bonds on the surface, and that leaves polar surface groups. So polar means that one side of the group is positively charged and the other is negatively charged. And that makes it water loving because water itself is polar. So when you put water on a hydrophilic surface, it spreads out because they bond to each other, they want to be touching each other. So that can help reduce water use because you need to use a lot less volume of water to cover the same amount of area or a larger area because it spreads out. It also helps because normally if you put water on a surface like this, it would bead up. And when it beads up, it drags the soiling into the middle, and then it'll dry and you'll end up with little patches of dirt like you get on your car or your windows if you've had heavy rain. So a hydrophilic surface also helps with that. It helps to spread it out and dry it more evenly. We've also discovered, which is really exciting, that plasma has a protective effect against soiling. So not only do we use less water when we do clean, but there's also something going on that means less soiling is sticking to that mirror as well. We don't yet understand why that is. So it's really exciting that I'm doing some research that perhaps no one else has done. I'm the first person that's finding this out, which is a great part of doing a PhD. So with those two things combined, we have the plasma having to remove the soiling. It helps to overcome the soiling bonds. We also have the modification of the surface, meaning less water covers more area. Plus, we're even protecting it from future soiling, which means we need to wash it less often anyway. So hopefully with my work, we aim to reduce the amount of water that's used and make these plants a lot more sustainable. So thank you for listening. If you have any questions, I'll gladly take them.
Great, thank you, Catherine. That sounds like a really cool application, and yay, plasma. Over to Natalia for some questions. I'm sure there are some.
Thanks, Barbara. Thank you, Catherine, for your work towards sustainability. And there are some questions. The first one is that plasma sounds a little bit dangerous with such a big amount of energy. Is it real? Is it that dangerous?
Yeah, so plasma comes in lots of different forms. So obviously something like this is very safe, you can even touch it. Most plasmas that are used in industry are, they're very hot, very high energy, and you wouldn't be able to use that on a mirror because it would crack it or melt it. So I use something that's called a non-thermal plasma. So the energy is all in the electrons rather than the heavier ions. So you can actually use plasma. They have some medical devices that use plasma. So you could, stick your hand under it if you wanted to. So you can get low temperature plasma, which is what I'm using. It does generate lots of UV sometimes. So you have to wear goggles, or in the lab, we have it in a special enclosure. So that's something that needs to be thought about as well. But kind of in this desert environment, there's not going to be lots of people standing around looking at it anyway. So for this application, it should be okay.
Okay, great. We have a question from Facebook. Luca is saying that this is very, very interesting. And he wonders if any plasma is found naturally on Earth.
Yeah, so lightning, as I said, is a form of plasma. So you have huge amounts of energy going from the sky to the Earth, and then that creates a plasma out of the air in that chain. You also have, the northern lights, the auroras, that's plasma as well. You also have it in consumer electronics, so plasma screen TVs. They're not as popular anymore because they're quite inefficient, but that uses plasma to generate lots of electrons, which then creates the red, blue, and green colors that you see. So that's another common form of plasma. Yeah, generally you wouldn't see that much plasma on a day-to-day basis, but it's mostly in space.
Okay, those were really good examples. We have a question from YouTube, from Facebook, from Julia. She didn't realize that lightning is an example of plasma. Does that produce UV light too?
Yeah, it probably does. But I mean, lightning doesn't hang around for very long, and you'd have to be quite lucky to be looking directly into it. But we have a lot of UV exposure to our eyes anyway. So most cataracts are actually caused by UV exposure. So if there is a lightning storm, you can look at it, that's okay. But I wouldn't recommend just staring into an electrical discharge.
Okay, that makes sense. We have another question. How easy is it to make a plasma in the desert instead of the lab, for example?
Yep, so I use a type of technology called dielectric barrier discharge. And it's a very robust technology because it's just an on-off operation. Some plasma torches are very kind of delicate. They have very precise conditions under which they operate. You have to tune them. It can take a lot of time. The advantages to that means you can have very energy efficient plasma. So you can have kind of 0.1% reflected power, so 99.9% efficiency. With my technology, it's not as efficient because you don't have any of those tuning capabilities. But it does mean that you can generate a plasma under pretty much any conditions. You can also generate it from the air, which is really important because you don't want to have to be buying lots of bottles of gas to use. You just need a compressor and a filter, and then you can just pull air straight from the atmosphere, push it through, and you'll get a plasma.
Okay, that sounds pretty interesting too. There's like a few questions around that. You've mentioned it a little bit, but maybe you can expand a little bit on it. If you need a lot of energy to produce plasma, because you've mentioned the efficiency, but maybe you can expand a little bit on that because there are some doubts around that.
Yeah, so it's really important that we don't save energy with the solar plant and then just use it to generate plasma. So we are working on increasing the efficiency. There are ways that you can make my technology more efficient. So my industrial sponsors just provided me with a new torch, which is more efficient. So it is important to have that balance between the energy that you generate through the solar power and the energy that you need to generate the plasma. So you're absolutely right. We need to make sure that we're doing it as efficiently as possible.
Okay, thank you very much, Katherine. It was really, really interesting, and good luck with your PhD. And over to you, Barbara.
Yeah, thanks. Yeah, thanks, Katherine. That sounds like I'm really fast. I have so many more questions. But we have to move on to our last speaker. And last but not least, we have Sana Ahmed, a PhD, a researcher in environment and waste management at the Open University.
Thank you, Barbara, for the introduction. Good afternoon, everyone. I'm Sana, and today I'm going to talk about food, specifically the food that we throw away. Remember that time you threw away your half-eaten sandwich or that bowl of leftover pasta that's been sitting in your fridge forever? And what about that long forgotten bag of salad waste that had to go straight from the fridge into your garbage bin? Yeah, we all have been there, done that. We are unknowingly contributing to one of the least addressed global issue, which is food waste. Globally, 1/3 of all food that we produce for human consumption is wasted, and that is worth over $1 trillion. However, alongside of gigantic amount of food waste, we have got enormous level of hunger. There are 800 million people in the world that goes to bed hungry every night. If we look here in the UK, we waste almost 10 million tons of food with a value of over 30 billion pounds per year. And it is believed that households are actually responsible for a larger portion of this. So what happens to this food waste after it throw away? Well, mostly we store in our household waste bins for a certain period of time, depending on local collection days, which is then collected by local authorities through refuse collection services and food recycling schemes. However, local authorities are encouraging householders to use kitchen caddies like this one to store food waste separately in kitchen caddies, which includes fruit, vegetable, meat, and other food remains, which leads to microbial decomposition of organic waste inside the kitchen caddy. as food waste has high moisture and nutrient contents, so it acts as an ideal feeding source for microbial growth. Therefore, the kitchen caddy can become an important source of various microorganisms, including bacteria, fungi, and airborne particles of variable biological origins called bioaerosols. So, do these microorganisms and the airborne components have any health effects on householders during storage and subsequently on waste collectors during collection of this waste? This is where my research comes in. My project aims to conduct a detailed audit of microbiological composition and bioaerosol emissions from household food waste during storage, collection, and treatment. To achieve this, I'm doing lab experiments to mimic real-world scenario where I'm generating and storing food waste materials in the lab under the same condition as food waste is produced in the UK homes. Then I'm taking both food waste and air samples from the bins to investigate microbial composition. So how do I determine what and how many microbes are present in these samples? Well, firstly, I'm taking samples and quantifying them in the lab by using selective growth media, which help me to isolate and quantify target microorganisms. This selective media actually act as food for microbes which have specific ingredients, to provide growth for microorganisms. For example, nutrient agar are used for bacterial growth, and malt extract is used for fungal growth. Then the next step is to identify these microorganisms genetically, genetically to get the more clear picture of their nature. For this, I'm using modern molecular techniques such as next generation sequencing. This requires extraction of genetic material like DNA and then amplifying the small segment of DNA called genes. In the end, I compare the resultant data with already available genetic databases to identify the microorganisms. By identifying these microorganisms, it would be possible to know whether they can pose health risks to householders or not. So, why is this knowledge so important? It is important to act when we know what sort of microorganisms and pathogens are present in the food waste that can lead us to identify the causative agent of health hazards. Ultimately, this study will provide information regarding how to minimize exposure to microbial emissions to householders when introducing an extended collection program, and will also help to develop the strategies aiming to mitigate or reducing the health risk associated with this. Thank you everyone for listening. I'm happy to answer every question.
Thank you, Sana. That sounds like, well, another fascinating topic with, I'm sure, many questions.
Thank you, Sana, for your talk. It was really, really interesting, also in a topic that it concerned all of us. So there are some questions for you. The first one is, what sort of bugs or microorganisms do kitchen caddies normally attract?
So, well, it depends on the nature of food waste. UK, a common household food waste, mostly of fruit, veg, salad, bakery, and kind of meat products and other many food products. So these food waste materials act as an ideal breeding source for various microorganisms, which include different kind of microorganisms, including bacteria, fungi, viruses, and other particles of various microorganisms. So my research is mostly focused on airborne microorganisms, because the bin has potential. Every time we open the bin and empty the bin, it has potential to release a microorganism into air, so we can get exposed to it. So the microorganisms I've been looking into, mostly they can cause serious gastrointestinal diseases and respiratory diseases. There are some microorganisms from Enterobacteriaceae family, like E. coli. I think all of you are familiar with E. coli. They can cause from light stomach cramps to even diarrhea and most severe stomach illness, and salmonella. And some molds are also very important in regarding my research, because the molds spread quickly into the bins, and they speed the rotting process of food waste into the bins. So the most frequently mold present in the bins is Aspergillus fumigatus, because they can cause serious illness like asthma and bronchitis and other respiratory diseases. So these are the microorganisms which is mostly concerned, recording my research.
Okay, that sounds a bit scary now. There is a question from Facebook from Emma, and she's asking, like, given these airborne pathogens that we can find, should we keep our food waste bins outside rather than inside the house? What do you think?
Well, like as you can say, when we prepare food, we just peel and other things and we put into the caddies. But in the normal household, kitchen caddies is almost 25 liters. So we put food into it and then we just leave it. But the best thing is it should have lid, and we should not touch we should not touch the lid of the bins, and mostly should be closed. So but we have to put inside the it's almost in our kitchen for at least for like 4 to 5 days until it fill. Then we can empty it into our outside bin. So I cannot just every time we put food waste into it, and every time we can go outside and put our food into outside. I think this is not acceptable practice, and we cannot do every time. We have to throw even a small thing like leftover sandwich, we have to put into the bin, and then it's in the bin. But at least we can empty the bin until it half full, or until at the end of the day we can empty it into the outside. But we have to put into the kitchen. This is the kitchen caddy. After finishing all the tasks, we can empty it into the outside. But it's fine for certain time period, but we cannot store for longer time period. And we can do other practices like other disinfectant and hygienic practices we can use to reduce the risk. Yeah.
Okay, thank you, because there was a question from YouTube asking how can we do to keep us safe from our kitchen caddies. I think you've answered that with keeping like taking them out regularly and disinfecting, right?
Yeah.
Okay, so there is just one last question. Well, there are more questions, but I don't think we have time for more here. But you've mentioned a lot of pathogens and dangerous airborne diseases. Are there any microorganism that could be good or can help prime our immune system, or are they always bad?
I'm afraid I have to cut you off, because okay. I know this is a really interesting discussion, but we have to keep in time, and I'm really sorry. As there have been some questions that haven't been picked up, we will make sure that the speakers will answer them in a blog post over the next couple of days. And we will post them below the live streams videos on our homepage. So make sure to check this out. And this will bring us to the end of this year's Soapbox Science Milton Keynes event. I hope you enjoyed the presentations from our fantastic scientists and hopefully learned something interesting about the research. I most certainly have, and I really enjoy it every time. I'd like to again thank all of our amazing speakers for their presentations, as well as for the people who joined to listen and ask questions. You're equally as important as the speakers. I'd also like to thank my fellow organizing committee members who have done fantastic work before the event as well as behind the scenes today. And a very special thank you to Katrina and Sam from the Open University, who, with their know-how as well as their help today in the background, made this event possible. And with this, I'd like to invite you to join us next year for another Soapbox Science event, and we will announce the format as well as the timing of that in springtime on our homepage and social media channels. So with this, I wish you a lovely Saturday and goodbye.